Let us continue our discussion on evolution of multicellularity. So, as we were discussing last time that we have a tube, the first phase of this experiment is only selecting for fast growth. After this fast growth, when this culture is occupied by yeast, we let the cells settle for some delta t amount of time, and in this time cells will have differential propensity to sink in the tube. Some cells will settle faster, some will float, and at this time I sample from the bottom of the tube and only transfer that 1:100 fraction to the next tube. And this selection pressure is important to understand, because what you are doing in this scenario is placing a hard constraint. At this point, what is being done is that if you settle fast, then you make it to the next round, and you make it to the next round and you stay in the experiment, right. If you settle fast, then you stay in the experiment. If however you are not settling fast, then you go extinct. And we never see these cells again in the evolution experiment. So, the selection pressure is a combination of fast growth and settling fast. What transpired in this experiment was that yeast cells have a transcription factor called ACE2, and a mutation was acquired in ACE2 such that this transcription factor, remember transcription factor controls transcription of other genes. So, this transcription factor controls expression of proteins of these genes, whose resulting proteins are responsible for. So, how cerevisiae divides is that it forms a bud, the bud then grows, and then finally some pinching off happens here and you get two individual progeny cells. But this final event of pinching off is dictated by several proteins, and the expression of these proteins is controlled by ACE2. What happened in this experiment was that the gene responsible for ACE2 picked up a mutation. So, a mutation happened here, as a result of which ACE2 was not functional or was not formed. Because there was no ACE2, the expression from these genes did not take place, these proteins were not formed, and as a result this pinching did not take place. And as a result, because these cells could bud, but these buds could not separate to become these individual unicellular cells, what we end up getting are these multicellular clusters. And there are beautiful images of this if you just search snowflake yeast, because they look like snowflake clusters. This was first discovered around 14 or to 15 years ago. Since then, at Georgia Tech, there is a lab which is run by William Ratcliffe, which has started an evolution experiment which starts with multicellular yeast and evolves this multicellular yeast. It tries to study what happens to this snowflake yeast cluster that I get if I evolve it for a really long time. What has been some recent results from that experiment is that as these clusters, so let me just draw this cluster. So, let us say this is a cluster, it's much bigger than this. I urge you to sort of just do a Google search of Snowflake East, and the images are quite stunning. So, as now this experiment has going on for a long time, and this experiment is around 10,000 generations have already been processed, starting with a multicellular cluster. Now, what is beginning to appear is that the cell at the center is sort of tasked with holding the entire cluster together. So, then its mechanical properties are quite different as compared to a cell at the periphery, which does not quite have the same mechanical stress that this central node of the cluster has to face. On the other hand, the central node has very little access to resources because it is just embedded in at the center of this cluster with cells all around. So, this has limited access to resources. The cells at the periphery however, have great access to resources. So, the access to resource is differentially spread over the cluster, and the mechanical loads, the mechanical demands that are being made up of different cells in the cluster are also differentially spread. The demand is much higher at the center of the node as compared to periphery, and access to resource is much higher at the periphery as compared to center of the node. So, what is beginning to be seen is patterns of differential gene expression, where the gene expression that this central cell is exhibiting has to do with greater mechanical strength and fewer protein expression which are responsible for bringing nutrients in. On the other hand, cells at the periphery, they are not, their gene expression is more to do with import of resources which will then be shared with other cells in the cluster, and has less to do with mechanical strength because its location in the cluster does not demand that. So, these are first signatures of diversification happening in a cluster which is where all cells are genetically identical. And this is important because this is what our cells are also doing. All of our cells are genetically identical. However, their manifestations are very different. A nerve cell looks very different from a skin cell to a liver cell, and so on and so forth. This is despite them having the same genetic content, and that is because the genes being expressed in one cell line are very different from genes being expressed in another cell line, and hence that differential gene expression gives the cells different properties, and hence they are able to sort of complete the respective tasks that they do in the body. And the signatures of the same thing are beginning to appear in this evolution experiment in Radcliffe's lab. But I also think that going forward in the years and decades to come, this experiment will also show some unexpected findings which will be of great value in understanding how evolution works. All right. The next experiment that we will be discussing has to do with how diversity is brought about. So, if I am starting with an isogenic population, suppose I have to design an experiment that starting from an isogenic population, what should I do such that this population splits into two or more groups, where one group of cells has picked up one type of mutation and the other group has picked up another mutation. Starting from an isogenic population and leading to this diversification process increases the diversity, genetic diversity in the population. This process is also called adaptive radiation. That I started with one type and it just diversified into many types and occupied all the different niches that were available in the environment that I provided the cell. This is obviously important from the context of understanding how niches are occupied. If I have how many species will be supported in a particular niche in an ecological setting. It is important from the process of understanding, from the context of understanding what are the processes and forces which generate biodiversity, because if you think of it starting from 1 and ending with 5, that is the process of generating diversity in species. So it's an important question from a variety of contexts, and I want to discuss two papers which have demonstrated generation of diversity in microbial populations. The first paper is by Rainey and Travisano in 1998. This is the paper not with E. coli, but with Pseudomonas. And what they do is they grow Pseudomonas in a test tube with a defined growth media. However, what they do not do is shake the tube, they just keep the tube static. So, no shaking. What this does is that it creates different niches available in a microcosm which is as small as a test tube. So, if there is shaking taking place, then each cell is being mixed and driven to different parts of the tube. Any gradient that might accumulate, so let us say that this part of the tube, if this is kept static, this part of the tube will have more oxygen available as compared to here. But if you are shaking it, so let us go back, if this is static, then this part, the top part will have more oxygen compared to the bottom part, and this establishes a gradient of oxygen. So, what it does, it creates niches here in a single test tube where you have one niche with low oxygen availability and you have another niche with high oxygen availability. Usually we are shaking these culture tubes like the flasks which are carrying the LTE populations are being shaken. But if you do that, you destroy that gradient and the whole environment inside the test tube is homogenized into this one setting. So, in that context there is only one niche, space is not a variable. In this context, when it is not shaking, space is a variable, because depending on my location, depending on my coordinates in the tube, the environment that I am seeing is quite different. So, if you think about this tube, there are three different niches available here. The first one is this, which is the air water interface. The second one is the glass surface, and the third one is bulk liquid. And starting with a population of isogenic Pseudomonas, if we propagate the population like this by just letting it sit and no shaking, very soon what we find is that the bacteria that occupy these three niches are genetically not distinct anymore. So, let us say these are three Pseudomonas populations, one sticking to the surface of the tube, second one floating in the bulk liquid, and third one at the air water interface. Each one of these niches is occupied by a unique genotype. And what was shown is that the number of mutations, so we can think of this as this particular genotype is the specialist at surviving at the interface. This one is a specialist in surviving in bulk, and this is the specialist which survives best at the glass surface. So, this adaptive radiation of just starting with a single genotype into these three specialist genotypes fairly fast was sort of is an important study in this context of understanding processes that lead to generation of diversity. However, what this does is that this studies diversity in an environment where there are three different niches which are available to the population. Can we create experiments where diversity is generated despite there being no separate niches? Suppose this tube was shaking, would we see that diversity? And the answer is no. People have studied adaptation of Pseudomonas in shaking flasks and shaking tubes, and there we do not see this diversity. So, the evolution of this diversity is contingent on there being present these three niches which are available to the population. So, the question that we ask for the next paper we discuss is, can diversity be generated in spatially homogeneous environments. So, the work that we just discussed has these three niches as we discussed: the air water interface, the glass surface, and the bulk liquid. But if this tube was shaking, then these three would not exist, because any individual, so the oxygen concentration would get homogenized because of the shaking, anybody sticking to the glass surface would be swept off by the convective currents, and so on and so forth. So, the question is that can diversity be generated in homogeneous environments? And this is sort of an area where our group has been working for a few years now. And the system that we work with is these disaccharides. So, let us consider a molecule of lactose which is comprised of glucose and galactose. Now, interestingly, glucose and galactose can be joined to each other via another way. So, another kind of bond can link glucose and galactose together. So, this square is now rotated 45 degrees and linked with glucose. So, this disaccharide is called mallebiose. E. coli bacteria can grow on both these sugars, and the way it grows is it grows on these sugars is that it first internalizes these sugars. So, the lactose goes inside, the same happens with mallebiose. Then we know that lac-Z acts on this, and what we get as the next step is hydrolysis of lac-Z inside the bacteria, and we get glucose and galactose released inside the bacteria for its consumption for growth and energy purposes. So, the key idea here is that this hydrolysis step, the disaccharide, whether be it lactose or mallebiose, is first internalized and then hydrolyzed. To monosaccharides. So, if that is the case, then what is present in the environment is only lactose or mallebiose, because this molecule is just internalized and then hydrolyzed inside the cell. However, yeast utilizes these disaccharides slightly differently, and we will discuss I will show you 3 different cases of how yeast processes these multiple monosaccharides which are linked differently. In the first case, the example is disaccharide which is called sucrose, which is a combination of glucose and fructose. And what happens in this case is that yeast does not internalize sucrose and then hydrolyzes it. In fact, yeast brings it to its periplasm and does hydrolysis here with the help of an enzyme called Suc2. The name is not important, but there is some enzyme which is encoded for in the yeast genome that it uses to hydrolyze sucrose without internalizing it, and the resulting monomers glucose and this is a triangle, glucose and fructose, 99% of them are diffused away in the environment. So, this is much over 95%, and only a few percent of them are retained by the cell for its own use. The rest of the monosaccharides which are released into the environment are available for everyone in the population, including itself. So, this creates a different dynamics as compared to an E. coli growing on these monosaccharides, because for E. coli the disaccharide is first internalized and then hydrolyzed. So, whatever is hydrolyzed, it's the product of hydrolysis are mine only, because I first internalized it and it was inside me that I hydrolyzed it. So, the glucose and galactose resulting from that process are only to be used towards my growth. That is not the case in this context, where sucrose is being hydrolyzed outside in the periplasm of the individual, and from there the resulting monosaccharides glucose and fructose, an overwhelming majority of it is released into the environment, and I only keep a very small fraction it for my own use. Now, that is not to say that this 95% is not available for my use, but it is to be equally shared between all members of the population that are surrounding me. The next case that is there in yeast is a sugar called mellibiose. Remember, mellibiose is glucose and galactose, and these two are linked differently than lactose. So, this is glucose, this is galactose. In this scenario, we have yeast which releases an enzyme. So, an enzyme which is encoded by a gene called MEL1, again the name is not important. This enzyme's job is to hydrolyze the mellibiose present in the environment into glucose and galactose. In the previous case, we saw that hydrolysis took place in the periplasm, and for its own use it kept 2 to 3% of the monosaccharides. In this case, even that is not there, the enzyme is being released into the environment and hydrolysis takes place in the extracellular environment. So, these monosaccharides that are released are available for anybody in the population. Right. This is an example of what is called a public good, where the resource does something which is for the benefit of the entire population. So, imagine that there is a species present here which cannot grow on mellibiose. So, if yeast is not there, then this species cannot grow because it can't utilize mellibiose. However, if yeast is present, it hydrolyzes mellibiose, leading to release of glucose and galactose, which can then be used by this species that is present. So, the question then is that why is yeast working towards such an inefficient scheme of utilizing mellibiose, where the work that it is doing to hydrolyze mellibiose into glucose and galactose is actually being reaped up, the benefits of that work are being reaped by other species and individuals present in the environment. And third is a sugar called raffinose. This is a link between galactose, glucose and fructose. And the way this is used is via a combination of the two strategies that we have already discussed. So, if we have this sugar in the environment, then MEL1 is secreted and MEL1 hydrolyzes this particular bond, which releases galactose and fructose. The fructose is brought to the periplasm and is hydrolyzed here, leading to release of glucose and leading to, this is sucrose. The sucrose is then brought to the periplasm, leading to hydrolysis of sucrose and release of glucose and fructose. Alternatively, raffinose can be brought to the periplasm and Suc2 acts on this, which leads to release of mellibiose plus fructose. This mellibiose can then be acted upon by MEL1, which leads to hydrolysis of mellibiose and release of glucose and galactose. So, the point being that there are these several public good type of systems in yeast, and our group has been using these systems to understand processes of diversification and how do populations interact with each other in the context of these public good systems. We will continue these discussions in the next video of the course.